Let us move on to the next of our formed elements. Remember that when you spin down blood, plasma rises to the top, the erythrocytes spin to the bottom, and then we're going to be taking a look at this guy right in here, which is called the buffy coat. So if we take that buffy coat apart and we look at what's inside of there, here we have basically this right in here is going to comprise your formed elements. Platelets, which we'll be talking about in the next video, and then leukocytes, which we're talking about in this video. Okay, so let's just open up the leukocytes and take a look at the different components of leukocytes. And what we'll find is that there's 1, 2, 3, 4, 5 basically different types of leukocytes that we work with. Leukocytes take up less than 1% of your blood volume, and again, we'll take a look at the typical white blood cell count for leukocytes. I want to talk about a few different terms that we want to kind of get out there and make sure that we're understanding them thoroughly, because students tend to these fall into the background and then they're not fully understood, or it's not really at the forefront of your memory when you're being assessed on this information. So the first term that we're going to be coming across is called leukocytosis. Leukocytosis is when your white blood cell count rises high, not hugely high, but it's elevated. It's an elevated level of leukocytes. And the reason why you undergo leukocytosis is that you're sick. You're sick. Your white blood cells are there to protect the body from damage, from external and internal insults. So when your white blood cell count is high and leukocytosis is occurring, it probably means that you're fighting off something that may be particularly nasty. The next term we're going to be coming across is diapedesis. Diapedesis is the concept that white blood cells can slip out of the capillary blood vessels and move through tissues and find stuff that they're interested in. The next term is called positive chemotaxis. Positive chemotaxis is when a white blood cell is able to follow a chemical trail released by, for example, damaged cells or other leukocytes, or perhaps some bacteria in the region or some kind of a pathogen. And the last of the terms is called amoeboid motion. This is actually how a white blood cell moves. It moves in sort of a blubby, jelly-like kind of a movement. Okay, so we'll take a look at some videos of that in class. Let's talk about our different types of white blood cells. Here, this is a nice figure because it actually shows the relative percentage of each of the white blood cells, each of the leukocytes. If you were to do just a specific white blood cell count, then the percentage you'd expect would be mostly neutrophils, secondarily lymphocytes, and then a small number of eosinophils, basophils, and monocytes. So let's take a look at those structures. What we have at the top here are the 3 granulocytes. Granulocytes are so called because inside of the cells you have these weird-looking nuclei, first of all, but ignore those, actually, not so important. What's most important is that you see granules inside of there, little punctate bits of material that take a dye up very well, so they tend to stain quite darkly. So granulocytes have membrane-bound granules. The granulocytes tend to be phagocytes, which means that they can consume other structures. They have kind of a tough life. They live only up to about 9 days, and they usually die fighting pathogens. The neutrophils, shown here, are the most numerous, about 50 to 70%. The neutrophils tend to fight off bacteria and some fungi. Eosinophils are next. Eosinophils take up about 2 to 4% of the white blood cell count, and they are specifically meant to fight off parasitic worms. Lastly, we have basophils here. Basophils are quite low in number. They tend to be about 0.5 to 1%. They contain histamine, which is a vasodilator. Sometimes they're so low in number that if you're doing a CBC, you might actually not find any basophils at all. Okay. The second, in the bottom row here, we have our agranulocytes. Agranulocytes, you put the word, the root A at the beginning of the word, it means without. So agranulocytes means without granules. There are 2 different types of agranulocyte white blood cells. The first is the lymphocyte. The lymphocyte, again, is a fairly large percentage, 25 to 40%, depends. They can survive a few hours, possibly, or they can survive for decades. It just depends on their role. You know lymphocytes better as T cells or B cells, or T lymphocytes and B lymphocytes. Usually these are found inside of the lymphatic system, which we'll be discussing down the line. The last of these that we're looking at here, the last of the agranulocytes, are called monocytes. Monocytes can actually live for several months, and you can see that they're actually quite large. They can differentiate into these much larger cells called macrophages. Macrophages tend to be about 18 micrometers in diameter, and the idea here is that macrophages are actually quite powerful structures. They really help to fight off disorders. We're going to be talking a lot about macrophages in the future. You may have actually heard of them before. Monocytes themselves do not in and of themselves fight off pathogens, but macrophages do. And a monocyte can differentiate into a macrophage within a couple of hours. The blood cell count for monocytes, the white blood cell count for monocytes is about 3 to 8%, depending. Taking a look at this nightmare, this is what we call leukopoiesis. So leukopoiesis is the differentiation of white blood cells. And you may notice something familiar here. When we look at the hematopoietic stem cell that differentiates into all of the different white blood cell all the different white blood cells, you start with a hemocytoblast. Do you remember where you've seen that term before? So the hemocytoblast is the structure that also differentiates into all of our red blood cells. Okay, so take a look at this. We'll be talking about this more in class, basically. You don't have to know all of the intermediary blood cells that occur here, but you do have to know the general concept. You start with a hemocytoblast, you differentiate into many, many intermediary cells, and then you eventually end up with the cells that we actually need. So eosinophils, neutrophils, basophils, there they are. We have our monocytes, we have our lymphocytes. Lymphocytes can further differentiate into other structures. We'll talk about that in great deal down the line. Remember that monocytes can differentiate into our macrophages, which are very powerful. Okay, so just kind of know the general concept of leukopoiesis and all of those intermediaries there. That's important for talking about the disorders that we'll be discussing.